So, um, if you look at the entire history of modern neurotechnology, there's this really recurring theme, like a sort of underlying obsession with brute force.
Right. Yeah.
We want to understand the brain, we want to interface with it, and our primary strategy for decades has basically been to drill a hole in the skull and shove a piece of metal into the tissue.
Exactly. It's very medieval when you think about it.
Right. We treat the human mind like like a malfunctioning motherboard that just needs a new wire soldered onto it.
Yeah.
And if you've been following the inescapable hype around brain-computer interfaces lately,
The BCI is making headlines every other week.
yeah, those. We are still doing it. We're making the probes thinner, sure, and making the robots that implant them faster, but the core philosophy remains completely unchanged.
It's a philosophy of conquest rather than collaboration. The prevailing assumption across the whole industry is that biological tissue is simply an environment to be managed.
Like it's a hostile territory.
Exactly, a hostile substrate. And the engineering challenge, as currently defined by the major players, is figuring out how to build a synthetic device that can just withstand that hostile environment long enough to extract a usable electrical signal.
But the paper we are unpacking for today's deep dives argues that this entire foundational philosophy is a dead end. We're looking at a piece of research by an independent researcher named Zachary David Enfield.
Titled The Organic Quantum Interface.
Yeah, and the central thesis here is going to completely upend everything you thought you knew about how human cognition can be linked to computation. I mean, Enfield is proposing a BCI that abandons the metal, abandons the silicon, and crucially abandons the surgery.
No wires penetrating the cortex whatsoever.
None. Instead, he outlines an architecture that communicates using the quantum biology of the brain itself.
Right, and what's so compelling about Enfield's approach is that he recognizes the ultimate goal of a BCI isn't simply moving a cursor on a screen.
Which is the benchmark right now, right?
Yeah, primarily because we are tapping into the motor cortex. But the true horizon of neurotechnology is semantic. It's about seamless, bidirectional interfacing with higher-order cognition.
Okay, let's unpack this. Because his argument is that you cannot achieve that level of semantic resolution by forcing a synthetic macro-scale electrical wire into a biological system that operates on, well, an entirely different set of physical principles at the nanoscale.
Exactly. You're trying to speak French to someone who only understands quantum physics.
Right. And I want to start exactly there with the failure of our current materials. Enfield calls this the crisis of the electrode paradigm.
It's a massive, sweeping critique.
Yeah, of every major BCI built to date, from the legacy Utah arrays to the highly publicized flexible polymer grids. He argues they all share a fatally flawed assumption, which is that a synthetic material can ever be made biologically acceptable to a living brain.
And to understand his critique, you have to look at the brain's immune system, which is incredibly aggressive. I mean, highly specialized.
Doesn't mess around.
Not at all. The brain has evolved over millions of years to maintain a pristine, strictly regulated internal environment. So the moment you introduce a foreign object,
Whether it's a stiff tungsten needle or an ultra-thin silicon thread,
right, you trigger an immediate cascade of neuroinflammation.
the dreaded glial scarring. It's literally the brick wall that every material scientist in this space eventually hits.
Precisely. The microglia, which act as the brain's, first responders, they detect the anomalous molecular signature of the implant.
They realize it's an invader.
Yeah, and they activate the astrocytes, which then undergo astrogliosis, essentially swarming the electrode. They encapsulate it in a dense web of scar tissue.
And that encapsulation physically pushes the neurons away from the recording sites, right? It creates this highly resistive barrier.
Exactly. Over months or years, the electrical impedance skyrockets, the signal-to-noise ratio plummets, and the device essentially goes blind.
And the fascinating part of Enfield's argument is that he views this not as a failure of our materials, but as a spectacular success of human biology.
The reactive oxygen species generated by this immune response are literally corroding the active electrode surfaces. The brain is doing exactly what it was designed to do.
Identifying an invader and neutralizing it. But the industry's response to this biological success has basically been to try and engineer a stealthier invader.
Oh, yeah. The last decade has been dominated by efforts to match the mechanical pliancy of brain tissue. Because silicon has a Young's modulus of around 200 gigapascals, it is rigid.
Super stiff. And cortical tissue is soft.
Very soft, sitting around 15 kilopascals. So, sticking a silicon array into the cortex is functionally equivalent to putting a jagged shard of glass into a bowl of gelatin.
Ouch. And with every heartbeat, the vascular pulsation causes the tissue to micromove, tearing against the rigid implant.
Right, so the engineers respond by developing polyethylene glycol dimethylacrylate hydrogels, or they use conducting polymers like ppGOT.PSS and flexible parylene C substrates.
Trying to bridge that massive mechanical mismatch. So basically, we keep trying to build a better, softer splinter, but the brain still knows it's a splinter.
A softer splinter is still a splinter, and that is Enfield's critical point. You can make the implant out of the most advanced, ultra-flexible, hydrogel-coated polymer on Earth.
Make it perfectly match the viscoelastic properties of the gray matter.
Right. But mechanical pliancy is merely a proxy for biocompatibility. It is not true molecular recognition.
What's fascinating here is that the microglia do not care how soft the material is, do they?
Not at all. They care that its surface chemistry does not speak the native aromatic protein-coded language of the central nervous system.
So we're trying to solve an immune crisis with a mechanical fix. If the brain is scanning for a specific molecular signature and we keep giving it synthetic polymers, we're always going to trigger the rejection cascade.
It wants a conversation partner, not a foreign object trying to hide.
Right. If we accept that premise, then the obvious follow-up is, well, what constitutes a native conversation partner?
And to answer that, we have to discard the classical model of neuroscience completely.
We have to look at how the brain actually processes information internally, deep beyond the macro-scale electrical firing we observe from the outside.
This is the first major paradigm shift in the paper. The classical model, the Hodgkin-Huxley model of the action, potential saying, views the neuron essentially as a wet, leaky electrical cable.
Electrical wave travels down the axon, hits the synapse, dumps some chemical neurotransmitters into the cleft, and triggers the next cell.
Yeah. And that model is the absolute bedrock of modern BCI design. We assume that if we can intercept that electrical wave, we are reading the mind.
But Enfield is saying it's an abstraction.
It's a huge abstraction. The classical action potential is undeniably real. But Enfield, drawing on a robust and growing body of quantum biology, argues that the action potential is merely the downstream output.
It's the printout.
Exactly, the printout. The actual computation, the integration of semantic meaning, memory, and cognitive intent is occurring internally within the neuron cytoskeleton, operating at a quantum optical level.
Okay, we are talking about microtubules now. And for anyone who has followed the fringes of neuroscience over the last 30 years, the mention of microtubules immediately brings up the Hameroff-Penrose-Orch framework.
Oh, absolutely. Stuart Hameroff and Roger Penrose proposed the orchestrated objective reduction theory back in the 1990s.
Suggesting that quantum vibrational computations in microtubules govern consciousness, right?
Yes, and for decades it was heavily marginalized by mainstream neuroscience. But recent advances in two-dimensional electronic spectroscopy and quantum biology have provided striking empirical validation for the underlying biophysics.
So what are microtubules, physically?
They're structural polymers composed of a protein called tubulin. They're about 25 nanometers in diameter, forming these cylindrical lattices that span the entire length of the axon and into the dendritic arbor.
And the molecular geometry of these tubulin proteins is where this gets incredibly specific. Enfield points out that every single tubulin dimer contains exactly 86 aromatic amino acids.
Yes, specifically 8 tryptophans, 36 tyrosines, and 42 phenylalanines.
And the spatial distribution of those aromatic rings is not arbitrary.
Far from it. The tryptophan residues, in particular, are spaced roughly 1 to 2 nanometers apart across the tubulin lattice.
And why is that nanometer scale spacing so critical?
Because it structurally mirrors the chromophore networks found in the light harvesting complexes of photosynthetic bacteria and plants.
Oh, wow. Let's pull on that thread.
Mm-hmm.
The Phennamathes-Olsen complex, the FMO complex in green sulfur bacteria. This was the watershed moment for quantum biology back in the late 2000s, right?
It really was. Researchers proved that these bacteria utilize quantum superposition to transport energy.
So in photosynthesis, a photon strikes a pigment molecule, creating an exciton, which is a bound state of an electron in an electron hole.
Right. And this exciton must navigate through a dense, chaotic protein matrix to reach a reaction center. If it moved classically, just bouncing around randomly, most of the energy would be lost as heat.
But instead, it exhibits quantum coherence. It essentially explores multiple pathways simultaneously. finding the most efficient route to the reaction center with near-perfect efficiency.
Yes. And the structure of the tryptophan network inside human microtubules is geometrically identical to those photosynthetic complexes. We literally have the same quantum hardware sitting inside our neurons.
But wait, here is the massive thermodynamic pushback, the critique that Hameroff and Penrose faced for years. I've always heard that quantum computers need to be kept at freezing temperatures.
Oh, the decoherence problem.
Right. They need temperatures near absolute zero and pristine vacuums.
Hmm.
Any environmental interaction, thermal vibration, electromagnetic noise, collapses the wave function. So how can quantum effects survive in a warm, wet, messy brain?
This is the thermal noise objection, and resolving it is fundamental to Enfield's architecture. The classical assumption is that thermal vibrations are random, chaotic noise that destroys fragile quantum states.
Right.
But the biophysics of phonon-assisted quantum transport turns that assumption completely inverted.
Phonons being quantized modes of vibrations occurring in a rigid crystal lattice.
Exactly. The tubulin protein is not a static structure. It is constantly vibrating. But because of the specific mass distribution and bonding within the protein sequence, these vibrations are highly structured.
So the thermal energy from the brain's environment drives these phononic oscillations.
Yes, and instead of destroying the excitonic coherence, these specific phonon modes actually match the energy gaps between the excitonic states in the tryptophan network.
Wait, really? The environment is actively pumping the quantum state rather than dampening it.
What's fascinating here is that the warmth of the brain isn't a bug causing decoherence, it is the vital feature actively sustaining it. The thermal phonons couple with the excitons, allowing them to hop efficiently along the aromatic network.
Like a perfectly timed push on a swing.
Exactly that. And this occurs in a fractal-like nested hierarchy of frequencies. The fundamental quantum dipole oscillations within the pi electron clouds of the tryptophan rings happen at the terahertz scale.
Microsecond, insanely fast optical processes.
Right. Those terahertz oscillations group together to drive gigahertz resonances across the microtubule lattice,
Which then modulate megahertz and kilohertz structural vibrations in the cell.
which eventually cascade all the way down to the classical hertz range.
Which is the millisecond classical action potential that we measure with an EEG or a Utah array.
Precisely.
So the classical electrical spike is just the macro-scale residue of a quantum optical computation that is already finished. We've literally been trying to read the receipt instead of the cash register.
That is the crucial takeaway for you, the listener. If you want to build an interface that genuinely captures cognition, you cannot rely on capturing the downstream classical exhaust. You must interface directly with the aromatic highway where the excitons are actively propagating.
And that brings us to the chemistry of the highway itself. We mentioned tryptophan and tyrosine earlier, but we need to understand why these specific amino acids are the primary conduits for these quantum states.
It's all about their electron configurations. Tryptophan is undeniably the VIP of this biological network.
Because it contains an indole ring system, right? Which is a bicyclic structure, a 6-membered benzene ring fused to a 5-membered pyrrole ring.
Correct. This large, flat, double ring structure provides an expansive surface area for pi electron resonance.
The pi electrons being the delocalized electrons that aren't tied down to a single covalent bond, they form these dense clouds of negative charge hovering above and below the flat plane of the molecule.
Exactly. And because of this geometry, tryptophan molecules can engage in face-to-face pi-p stacking.
Like a stack of microscopic pancakes.
Perfect analogy. When these flat indole rings stack parallel to one another, their delocalized electron clouds overlap. This orbital overlap creates a highly efficient continuous electronic corridor.
So an excitant can migrate down this stacked column with minimal resistance.
Yes. Tyrosine, on the other hand, contains a single phenol ring with a hydroxyl group. It has a different electrostatic profile and prefers a T-shaped stacking geometry, where the edge of one ring points toward the face of another.
So if you look at the microtubule architecture, the tryptophan provides these ultra-fast parallel conduction columns running actually down the tube.
While the tyrosine provides the lateral T-shaped crosslinks between those columns, it forms a robust two-dimensional quantum optical mesh, not just a one-dimensional wire.
But the true elegance of this system is how it interacts with the extracellular environment, right? Because the classical synapse and this internal quantum microtubule network are not parallel, isolated systems.
No, they are physically and electrically coupled together. If we connect this to the bigger picture and look at the research into cation-pi interactions by scientists like Dennis Doherty, we see exactly how this transduction occurs.
Cation-pi interactions. We're talking about the electrostatic attraction between a positively charged ion, the cation, and the dense, negatively charged pi electron cloud of an aromatic ring.
Right. Most major neurotransmitters, acetylcholine, dopamine, serotonin, are cationic at physiological pH. They carry a positive charge.
So when acetylcholine crosses the synaptic cleft and binds to its receptor on the postsynaptic neuron, it isn't just fitting into a generic physical pocket.
The binding event is fundamentally driven by a cation-pi interaction. The positive charge of the neurotransmitter binds directly to the pi electron clouds of the tryptophan and tyrosine residues lining the receptor site.
And those receptor residues are physically contiguous with the internal microtubule cytoskeleton.
Yes. The binding of the neurotransmitter pulls on the electron cloud of the receptor's aromatic rings, which instantly perturbs the coupled pi-electron network of the underlying microtubule.
So that single chemical binding event is the exact transduction mechanism that converts an extracellular classical chemical signal into an intracellular quantum optical state.
The chemical drop causes a quantum ripple across the entire tubulin mesh.
That reframing completely changes how we approach neurotechnology. If the thought process is happening as an excitonic ripple through a tryptophan network, then any interface we build must be able to speak that exact language.
Right. But before we get to the material science of building that device, we have to talk about location. Where in the brain do we actually try to tap into this network?
Because current BCIs are almost exclusively targeting the primary motor cortex.
The motor cortex is a highly accessible target, and decoding motor intent is vital for restoring mobility to paralyzed patients. But anatomically and functionally, the motor cortex is simply the output terminal.
The signals there are essentially finalized commands. Move this muscle fiber at this specific vector.
Exactly.
It's like intercepting a letter that has already been written, sealed, and dropped into the mailbox. You can read the address, you can see where it's going, but you have no insight into the complex formulation of the ideas contained inside.
That's a great way to put it. Enfield argues that if we want semantic resolution, if we want to decode the abstract ideas, the imagined words, the working memory we have to target the prefrontal cortex.
And the anatomy of the PFC is wild. You've got the dorsolateral for cognitive control, ventromedial for value-based decisions, anterior cingulate for conflict monitoring.
It is the anatomical seat of executive function. This is where the synthesis of sensory input, memory retrieval, emotional valuation, and forward planning coalesces into a decision before a motor command is ever drafted.
Reading the prefrontal cortex is like reading the author's mind as they decide what to write. But targeting the PFC presents a massive anatomical hurdle.
Because the computational heavy lifting in the prefrontal cortex happens deep within the cortical layers.
Right. If you want high-resolution signals from those regions, you typically have to drill through the skull, plunge a multi-electrode array centimeters deep into the tissue, and trigger the exact glial scarring immune crisis we discussed earlier.
That is the standard assumption. However, Enfield highlights an extraordinary anatomical loophole specific to the human prefrontal cortex.
Oh, I love a loophole.
The most computationally significant neurons in the PFC are the massive layer V and layer 3 pyramidal neurons. Their cell bodies are indeed situated deep within the cortical column.
But they project these heavily ramified, elongated structures called apical dendrites.
And these dendrites act as the primary listening posts for the neuron, integrating inputs from thousands of other synapses.
And in the human PFC, these apical dendrites exhibit profound elongation. They stretch vertically 2 to 3 millimeters all the way from the deep layers up to layer I.
Immediately beneath the pile surface.
The pile surface being the ultra-thin outermost membrane of the brain tissue.
Right. And once they reach layer I, they arborize, they branch out into a massive, dense canopy of dendritic tufts.
And these apical dendrites are not passive cables just relaying a signal down to the cell body, right?
Whoa. They are active, highly complex computational compartments, and they are densely packed with the exact quantum-active microtubule networks we've been analyzing.
The implications of that are staggering for you, the listener. The brain has essentially built a high-bandwidth access port right at the surface. You don't need to penetrate the gray matter at all.
The highest density of quantum-active excitonic processing structures involved in deep executive thought extends to within micrometers of the outer surface of the brain.
So if we can place a sensor in conformal contact with the pile membrane, We bypass the penetrating trauma entirely.
We bypass the trauma, yes, but we still face the immune recognition issue. Even resting on the surface, a synthetic polymer will eventually provoke a meningeal inflammatory response.
Right, so we need a material that can sit on the pile surface, speak the quantum optical language of the underlying dendrites, and completely evade immune detection.
Which brings us to Section 5, and arguably the most provocative, potentially alarming part of Enfield's proposal. To build this biological conversation partner, he turns to synthetic biology.
He proposes engineering an amyloid scaffold derived from a yeast prion protein called SUP25. And I have to stop here because the moment you introduce the words amyloid and prion in the context of human neurology, every single alarm bell starts ringing.
I completely understand.
When I hear prions, I think of mad cow disease or Creutzfeldt-Jakob. When I hear amyloids, I think of the toxic plaque buildup in Alzheimer's disease. Why on earth would we deliberately put that on a human brain?
The visceral reaction is entirely understandable, but it stems from conflating human disease pathology with the broader structural biology of these proteins.
Okay, so distinguish them for me.
It is true that human amyloids, like the amyloid beta peptide in Alzheimer's or the misfolded PRP protein in prion diseases, are neurotoxic. They induce a cascade of conformational changes in native proteins, leading to massive cell death.
Right, which is terrifying.
But the SUP35 protein is a yeast prion. It is native to Saccharomyces cerevisiae, common baker's yeast.
And in yeast, it doesn't cause a disease.
Not at all. In yeast, SUP35 is a functional, heritable epigenetic element. The amyloid conformation is a natural, biologically healthy state that allows the yeast to rapidly adapt to environmental stress.
Okay, but it's still a prion.
But the crucial factor here regarding safety is the species barrier. The sequence of the SUP35 prion is fundamentally absent from the mammalian proteome.
So it's a completely foreign protein. But wait, if it's foreign, won't the immune system recognize it as an invader? We just spent the first half of this deep dive establishing that the brain attacks anything it doesn't recognize.
This is the genius of the biological Goldilocks zone Enfield is targeting. Prion infectivity and amyloid cross-seeding are highly sequence-specific.
Meaning they have to match.
Exactly. A yeast prion cannot act as a template to misfold a human protein because the amino acid sequences and the resulting steric geometries do not match. It is biochemically impossible for SUP35 to trigger Alzheimer's or Creutzfeldt-Jakob in a human brain.
So it's entirely safe from a pathological standpoint. It won't cause a misfolding cascade. But why doesn't the immune system attack it for being foreign?
Because while it is sequence novel, it is structurally native. The immune system's pattern recognition receptors are primed to detect the rigidity of silicon, the unnatural surface chemistry of polymers, or the specific molecular patterns of bacterial cell walls.
But the SUP35 scaffold is constructed entirely of natural biological amino acids, folded into a standard cross-beta sheet structural motif.
And the human brain contains hundreds of non-pathological functional proteins that utilize beta sheet architectures. To the microglia, this engineered scaffold simply registers as inert biological tissue.
It slips right past the inflammatory triggers because it speaks the ambient chemical language of biology. It's a molecular Trojan horse.
Precisely.
But is a protein scaffold tough enough for a long-term implant? Brains move. Fluid washes over them. Won't a yeast protein just dissolve or degrade over a few months?
The cross-beta sheet spine of an amyloid fibril is one of the most thermodynamically stable and mechanically robust protein assemblies in nature. It is a quasi-crystalline structure, heavily stabilized by a dense, continuous network of intermolecular hydrogen bonds.
How strong is it exactly?
Its mechanical stiffness measured in Young's modulus ranges between 3 and 20 gigapascals.
That is incredibly dense. That puts it in the same structural class as silk or engineered nylon.
It is functionally permanent within the physiological parameters of the brain. But Enfield doesn't just propose using the natural SUP35 protein. He proposes heavily augmenting it.
Right.
He targets the specific octopeptide sequence responsible for fibril formation and genetically engineers it.
This is where we bring back the tryptophan and the tyrosine, the quantum highway.
Exactly. The geometry of the beta sheet dictates that adjacent amino acid strands are spaced exactly 4.8 angstroms apart along the length of the fibril.
So by engineering the genetic sequence to substitute a synthetic tryptophan residue at precise periodic intervals.
Specifically, one per turn of the beta strand.
Those large aromatic indole rings line up perfectly. They stack face to face, just like the pancakes we talked about earlier, and they are locked into a spacing of exactly 4.8 angstroms.
And that 4.8 angstrom spacing falls flawlessly within the optimal 3.5 to 5 angstrom theoretical window required for robust pipey stacking and highly efficient quantum coherent conduction.
We are deliberately directing the self-assembly of a synthetic biological quantum wire.
Yeah.
Here's where it gets really interesting, though. We aren't just using yeast proteins. We are augmenting them with inorganic nanotechnologies, biomineralization.
Right. The scaffold is structurally augmented during the self-assembly phase to boost its transduction capabilities. The most critical addition is the incorporation of nanodiamonds containing nitrogen vacancy, or NV, color centers.
Nitrogen vacancy centers.
Yeah.
These are atomic scale defects where a nitrogen atom replaces a carbon atom in the diamond lattice sitting next to an empty space. And they're exceptionally sensitive to magnetic fields, right?
They're arguably the most sensitive room temperature quantum magnetometers known to physics. The electron spin state of the NV center is highly responsive to minute fluctuations in local magnetic flux.
So by intercalating these nanodiamonds into the amyloid matrix, the scaffold gains a secondary independent readout channel to detect the magnetic signatures of the quantum states propagating in the brain's microtubule.
Furthermore, Enfield proposes embedding graphene nanoribbons between the fibril layers to enhance long-range electron delocalization and utilizing heavy metal uranyl binding motifs to modulate the overall electronic band gap of the aromatic columns.
It's just an astonishing convergence.
Yeah.
You have a biologically synthesized yeast prion backbone, perfectly engineered with a custom tryptophan quantum wiring system studded with nanodiamond sensors and graphene ribbons.
It's beautiful engineering.
It sits seamlessly on the pile surface, structurally robust, completely invisible to the immune system, and perfectly tuned to the quantum optical language of the apical dendrites beneath it. But this incredible organic device still has to function as a computer.
True.
It has sensors, it has to process data, and it has to transmit that data out through the skull, which means it needs power. And remember, we aren't using wires or batteries.
Power delivery is the Achilles heel of almost all chronic neural implants.
Because if the core mission is zero penetrating wires and zero surgery after implantation, you cannot rely on an external battery pack plugged into a port in the skull.
No, you can't.
And if this device is intended to sit gently on your brain for 20 or 30 years, you certainly aren't scheduling a craniotomy every 2 years to swap out a degraded lithium-ion battery. So where does the juice come from?
Enfield's solution is a trimodal ambient energy harvesting architecture. It relies on 3 continuous independent ambient energy sources natively present within the cranial vault.
The first mode he proposes is beta-voltaic energy harvesting. And I have to push back here again, just like I did with the prions. A beta-voltaic cell relies on radioactive isotopes.
Yes, it does.
He is explicitly suggesting we incorporate radioactive material tritium, or carbon-14, directly into the structural backbone of an implant sitting on the human cortex. The optics of that are terrifying, and the biological risks of radiation-induced DNA damage seem entirely counterproductive to the goal of a safe interface.
The apprehension is logical, definitely, but it requires a strict understanding of particle physics, specifically the stopping power of beta radiation in biological tissue. We are not discussing gamma radiation, which is highly penetrating and destructive.
Right, gamma goes through walls.
Exactly. We are discussing beta decay, where an unstable nucleus emits an electron, a beta particle. Tritium, for instance, has a half-life of 12.3 years and emits a beta particle with an average energy of only 5.7 kilo electron volts.
How far can an electron with that energy travel through water or brain tissue?
The maximum penetration depth of a tritium beta particle in liquid water is approximately 6 micrometers.
6 micrometers. Wait, a typical pyramidal neuron cell body is like 15 to 30 micrometers across?
Exactly. The beta particle lacks the kinetic energy to traverse even a single cell. The beta voltaic elements are integrated intimately into the nanometer scale layers of the scaffold itself.
So 100% of the kinetic energy of the emitted beta particles is deposited entirely within the local matrix of the device?
It never reaches the underlying pile surface, let alone the genetic material of the neurons.
The shielding is just the inherent density of the microenvironment, so it's effectively a microscopic, entirely self-contained nuclear battery.
Yes. The power yield is exceptionally low on the order of nanowatts to single-digit microwatts, but it is absolutely continuous, unaffected by the physiological state of the host, and persists for decades.
It provides the unbreakable baseline power for the interface's deepest analog logic circuits. Okay, radiation makes sense for a baseline trickle of power, but the brain is dynamic.
Highly dynamic.
If a user is suddenly engaged in intense cognitive load, complex spatial reasoning, or emotional processing, a constant nanowatt trickle might not scale fast enough to handle the spike in signal transmission. Is there a way to harvest burst energy from the brain's actual real-time physical activity?
That leads directly to the second mode, piezoelectric harvesting. The brain is not a static organ resting quietly in the skull. It is a highly vascularized, mechanically dynamic structure constantly bathed in cerebrospinal fluid.
Right.
With every single heartbeat, the systolic pulse wave propagates through the cerebrovascular tree. This hemodynamics produces a rhythmic physical displacement of the entire cortical surface at approximately 1 to 2 hertz.
The brain physically swells and pulses with the heartbeat, and it also moves with respiration. Right.
Yes, the intrathoracic pressure changes during breathing translate into the cranial vault, causing slower, sustained pressure oscillations at 0.2 to 0.4 hertz.
So the cortical surface is constantly undergoing microdeformations, expanding and contracting by tens of micrometers.
And piezoelectric materials possess a unique crystallographic structure where physical mechanical stress squeezing or bending displaces their internal charge, generating an electrical voltage.
And the engineered amyloid beta sheet structure intrinsically exhibits some piezoelectric behavior.
It does. However, to maximize efficiency, Enfield proposes embedding highly efficient inorganic piezoelectric nanoparticles, such as barium titanate or lead zirconate titanate, into the flexible hinges of the scaffold matrix.
So every time the host's heart beats, every time they take a breath, the tissue gently squeezes the scaffold, and the scaffold transduces that mechanical strain into electrical bursts. That is profoundly elegant. Your own heartbeat provides the dynamic power for the interface reading your thoughts. And what is the third mode?
The third is thermoelectric harvesting, which capitalizes on the thermal gradient across the cranial vault via the Seebeck effect.
The Seebeck effect occurs when you have a temperature differential across a conductive material, which drives the diffusion of charge carriers from the hot side to the cold side, creating a steady voltage.
The brain is arguably the most metabolically demanding organ in the human body. Despite accounting for only 2% of total body mass, it consumes roughly 20% of the body's total energy expenditure. Consequently, it runs hot.
The deep cortical tissue and the pile surface are highly thermogenic, typically maintaining a temperature around 36 to 37 degrees Celsius.
But the environment directly above the device, the skull, the scalp, and the ambient air is significantly cooler. The scalp surface is usually between 30 and 35 degrees, heavily dependent on external room temperature.
Therefore, a permanent continuous temperature gradient of 2 to 7 degrees exists precisely across the physical gap occupied by the interface.
By integrating nanoscale thermoelectric generators, using bismuth telluride or similar alloys into the superior aspect of the scaffold, the device continuously extracts an electromotive force from the sheer metabolic heat of the brain's operation.
It is the ultimate closed-loop system. The device on your prefrontal cortex is powered infinitely by a microscopic fraction of atomic decay, the rhythmic mechanical pulse of your cardiovascular system, and the literal metabolic heat generated by your deepest thoughts. Trimodal ambient harvesting.
No external batteries, no penetrating wires, no thermal degradation.
And crucially, these three modalities feed into a shared nanoscale supercapacitor within the device, smoothing out the pulsatile mechanical power and integrating the constant baseline flows into a stable energy supply for the logic and transmission layers.
Which brings us to the architecture of the device itself.
Right. How do all of these molecular and energetic components stack together to actually process a signal? Enfield outlines a 6-layer ambient architecture that sits between the pile surface and the skull. We need to walk through the journey of a signal, from the moment a quantum excitant fires in the brain to the moment it becomes readable data on the outside.
The journey begins at layer 1, the quantum coupling layer. This is the base of the amyloid scaffold, sitting in direct conformal contact with the pile surface.
And its singular critical function is to link its own synthetic tryptophan network with the biological tryptophan network inside the apical dendrites, resting just micrometers beneath the pia.
And it has to do this without physically penetrating the membrane. It has to link without touching.
How does it do that?
It achieves this via Förster resonance energy transfer.
Let's really break Fabert down, because this is the core mechanism of the entire paper. If I have 2 tuning forks that are perfectly calibrated to the exact same pitch, and I strike one so it begins to ring, and then I bring it into close proximity with the second, silent tuning fork, the second one will begin to vibrate and ring on its own. The acoustic energy transfers across the gap without any physical contact between the two forks, simply because they share a resonant frequency.
That is the exact conceptual framework, but applied to quantum excitonic states rather than acoustic waves. In FRET, we are dealing with transition dipoles.
Okay, transition dipoles.
When an exciton propagates down the biological microtubule in the dendrite, it creates a fluctuating electromagnetic field, a dipole oscillator. If the synthetic tryptophan ring is in the scaffold, are engineered to have an overlapping emission and absorption spectrum.
If they are tuned to the same pitch.
Exactly. They engage in dipole coupling. The excitonic energy non-radiatively transfers across the pile membrane directly into the scaffold's quantum network.
But Enfield notes a severe physical constraint here. The efficiency of FRET is inversely proportional to the sixth power of the distance between the donor and the acceptor.
Yes, the inverse sixth power law. It means the energy transfer efficiency drops off an absolute cliff the moment the distance increases even slightly.
So it only works super close up.
FRET is functionally non-existent beyond 10 nanometers. This is precisely why targeting the prefrontal cortex's apical dendrites is the only way this architecture works.
Because those dendritic tufts arborize immediately beneath the pia, the physical gap between the biological microtubule and the synthetic scaffold is compressed to between 2 and 10 nanometers.
Within that ultra-narrow sub-10 nanometer window, FRET efficiency is massive, reliably transferring 50 to 80% of the quantum state.
The thought literally leaps across the boundary. And because FRET is a bidirectional physical process, this isn't just a listening device. If the scaffold generates its own excitonic state, it can FRET couple back into the brain. It can read and it can write.
Precisely. As the quantum signal propagates upward into the device, it reaches layer 2, the biomineralization layer.
This is where the nanodiamond NV centers continuously monitor the magnetic flux of the excitonic transfer, providing a parallel data stream to cross-verify the integrity of the FRET signal.
This layer also harnesses spontaneous biophoton emissions.
Biophotons. Wait, the brain produces light.
It does. Normal oxidative metabolism in neurons produces ultra-weak, spontaneous photon emissions, typically in the 400 to 800 nanometer visible spectrum.
I had no idea.
It is incredibly faint, but layer 2 utilizes highly sensitive organic photovoltaic elements to capture these biophotons, both as a secondary proxy metric for metabolic activity and to squeeze out a marginal addition to the energy harvesting pool.
Then the signal passes into layer 3, the piezoelectric signal conditioning layer. We talk about how this layer harvests power from the heartbeat, but it also has a data function.
When a neuron fires a classical action potential, the massive influx of ions causes the cell membrane to physically swell. It bulges outward by a few nanometers.
And layer 3 is sensitive enough to detect these transient nanoscale pressure signatures.
This provides the interface with a classical electrical readout, temporally correlated with the quantum data. We get the deep excitonic cause and the subsequent macroscale mechanical effect.
The signal then hits layer 4, the nanoscale power management and logic layer. This is the actual integrated circuit chip of the device.
It manages the complex power routing from the trimodal harvesters, but its primary computational role is data compression.
Because the raw analog quantum signal coming up from the FRET layer is far too dense to transmit raw.
Exactly. The IC chip encodes these continuous analog wave functions into discrete spike timing event streams. It turns the complex wave into a highly compressed digital Morse code.
Which prepares it for layer 5, wireless near-field communication.
An integrated gigahertz microantenna built directly into the perimeter of the scaffold wirelessly transmits this compressed data stream straight through the intact skull.
Eliminating the transcutaneous wire entirely negates the massive infection risks that plague current semi-invasive neurotechnologies.
And finally, layer 6. The signal reaches a wearable receiver sitting quietly on the user's scalp.
The external AI decoding layer. This wearable module houses sophisticated transformer-based neural network decoders.
Because the signal originating from the quantum array layer is so temporally pristine and possesses single dendrite spatial resolution without the muddying artifacts of traditional EEGs or the inflammatory noise of penetrating electrodes, the AI can decode the spike timing stream with unprecedented accuracy.
We have walked through the molecular biology, the synthetic engineering, the nuclear and thermodynamic power harvesting, and the entire transmission stack. But this brings us to the ultimate question for you, the listener. The so what? What does this miraculous, non-invasive quantum architecture enable us to actually do that a Neuralink cannot?
It fundamentally alters the semantic resolution of the interface.
Let's define semantic resolution in this context.
When current BCIs record the motor cortex, they are capturing the final compressed output of a decision. They decode the intention to flex a bicep or extend a finger. Semantic resolution refers to the depth of meaning we can extract. Because the Enfield architecture interfaces directly with the quantum computational layer of the prefrontal cortex, the anatomical hub of working memory and executive control, we bypass the motor output entirely.
We are not reading twitches. We are reading the cognitive representation itself.
We are capturing the excitonic states that correspond to imagined words before they are ever vocalized. We are decoding spatial models as the mind internally visualizes navigating a room. We are intercepting the formation of abstract categories.
We are reading thoughts in their raw native format. But the implications extend far beyond simply controlling a computer or a robotic prosthetic with unprecedented fluency. If the device can read at that semantic depth, and crucially, if it can write back into the microtubule network via reverse FRT coupling.
The therapeutic medical applications must be staggering.
Exactly.
Consider the current state of neuromodulation for severe refractory psychiatric conditions. Major depressive disorder, treatment-resistant obsessive-compulsive disorder, PTSD, schizophrenia.
Current interventions, whether pharmacological or device-based like transcranial magnetic stimulation or deep brain stimulation, are incredibly blunt instruments.
They really are. They bathe the entire brain in chemicals or subject massive swaths of tissue to macro-scale electromagnetic fields.
It's like trying to tune a piano by hitting the entire keyboard with a sledgehammer.
But computational psychiatry has demonstrated that these disorders are fundamentally rooted in highly specific, localized dysregulations of prefrontal circuit dynamics. They are driven by pathological attractor states.
Attractor states, a dynamic system naturally pulling toward a specific pattern, like a deep groove worn into a vinyl record. The needle just keeps slipping back into that same looping scratch.
That is the perfect visualization. An intrusive, looping catastrophic thought in OCD, or a sudden, uncontrollable limbic flashback in PTSD, is a pathological attractor state. The quantum excitonic dynamics of the prefrontal cortex get violently pulled into an inescapable loop.
So what does this all mean? Walk me through a PTSD intervention millisecond by millisecond.
Because the interface operates continuously at the quantum layer, it detects the specific excitonic signature of the pathological attractor state, the precise microsecond it begins to form in the apical dendrites.
It detects the groove before the classical electrical action, potentially even fires. Before the user consciously experiences the dread, the physiological arousal, or the visual flashback.
It sees the thought before it reaches conscious awareness.
Wow.
Instantly, the Layer 4 logic chip calculates the precise inverse perturbation required. It sends a signal to the Layer 1 scaffold, which emits a targeted, highly specific FRT signal back across the pile membrane, directly into the affected dendritic microtubule.
It injects a quantum optical perturbation that destabilizes the pathological excitant,
Bumping the neural dynamics out of the groove and seamlessly guiding the circuit back into a healthy baseline computational state.
correcting a PTSD flashback at the quantum level in real time without the user ever feeling it. It is genuine closed-loop cognitive neuroprosthetics. It actively modulates and heals the computational substrate of the mind.
And Enfield explicitly outlines a 6-priority research agenda to achieve this, noting that the requisite technologies already exist across siloed disciplines. We don't need to invent new physics. We need deep interdisciplinary integration.
We need synthetic biologists to synthesize the tryptophan-inserted sub-35 fibrils. We need quantum physicists to use 2D electronic spectroscopy to validate the FRT coupling between those fibrils and isolated biological microtubules.
We need material scientists to build the tissue phantoms to prove the trimodal energy harvesting works continuously under biological constraints.
The quantum biologists, the synthetic protein engineers, the nanodiamond specialists, they just need a unified architectural goal. We need to stop drilling holes and start building bridges.
Precisely.
We started this discussion with the image of that rigid, static engineering paradigm, the metal splinter, the brain scarring over it, fighting our attempts to conquer it with brute force. And over the last hour, we have traversed all the way to a deeply elegant, self-powered, biologically native quantum scaffold.
It's an incredible journey.
A device that rests gently on the pile's surface, fueled by your own heartbeat and body heat, whispering to the prefrontal cortex in the quantum excitonic language it has spoken for millions of years.
It is a profound shift in perspective. It represents a necessary maturation in how we approach neurotechnology, moving from an ethos of domination to one of deep biological symbiosis.
To collaborate with the mind at the deepest possible level. But as we wrap up this deep dive, I want to leave you, the listener, with a final, slightly provocative thought to mull over. We spent a lot of time exploring how this bidirectional interface can seamlessly read your working memory, and crucially, how it can write delivering precision quantum perturbations to instantly correct pathological loops like severe depression or trauma response.
Yeah.
But if we successfully construct a machine that perfectly reads and writes excitonic quantum states directly into the absolute core of your executive functioning, if this ambient interface is constantly silently correcting your thoughts at the microsecond level, smoothing out the rough edges of your cognition before you even consciously process the emotion, where exactly does the device end and where do you begin?
That is the question.
If a synthetic quantum network is continuously shaping your decisions to keep your mind perfectly healthy and optimized, what happens to your fundamental sense of free will? Who is actually making the choice, the organic brain or the organic interface? Thank you for joining us on this deep dive. Keep your mind open, stay immensely curious, and we'll catch you on the next one.